Saturday Morning Physics is sponsored by the M. Lois Tiffany Endowment, the Hideko Tomazawa Endowment, the Physics Department, and by gifts from friends of the program.
I'm like everybody else. I started my career thinking geological time is long, Earth history is long, and evolution is slow. And I guess the first thing that struck me as I was a student and then became a professor is how little discussion there was about how fast is evolution. And slowly, through a series of empirical studies, I got interested in this question and want to tell you about it today. This is a nice occasion to speak because I'm going to talk about Darwin. It's the 200th anniversary of Darwin's birth. It's the 150th anniversary of the publication of The Origin of Species. And so it's a real Darwin year this year. Secondly, this is a museum year. On campus, we have a museum theme year emphasizing the research and the exhibitions that go on in the museums on campus, and we're very lucky at the University of Michigan to have really strong research museums and excellent exhibit facilities too. And then finally, I'm going to talk about evolution, and there's a sense in which evolution is for all time. It helps explain the past. It's happening now, and we can be confident it will continue in the future. So with those three themes in mind, I want to start here. We've known that geological time was long for about 200 years. And the evidence comes from the sequence of strata. This is an illustration from Alexander von Humboldt, reproduced by Georges Cuvier in 1825, showing the principal stratigraphic layers of the European basins with what was called then the Tertiaire Intermédiaire. And here's Paleozoic with trilobites in it. The Secondaire, which is what we today call Mesozoic, and the Cenozoic up here with mammals like whales and primates and the groups that we're familiar with in it. The geological time scale is divided into a Paleozoic, a Mesozoic, and a Cenozoic because it's all about the zoo. It's all about animals. And the sequence in which they're stacked up in time is the same all over the world. Now, there are details in smaller regions that differ from other regions, but the general sequence through time is the same. And we've known this for 200 years. Interestingly, the part of evolution that creationists disagree with is the part that's stacked in stone in the succession of faunas up through time. There's no way to get around it. It's there. Now, how to put numbers on this is more difficult. It's one thing to have a stack of fossils up through time. It's another thing to have any idea how old the old ones are. And Charles Lyell's contribution to this, and this again goes back to the 1830s. Here's a photo of him, or a drawing of him in 1836. He'd already published 3 volumes of the Principles of Geology here by the time this drawing was made. He figured out that you could explain most of what we see by processes acting today if you assumed enough time. He couldn't put a number on it. He just basically assumed that the Earth was as old as it needs to be. Evolution was as long as it needs to be to explain what we see. And this is the context in which Darwin did his work. And I like this illustration, Charles Darwin, geologist, because I'll remind you, he was a geologist until he published The Origin of Species. And then everything he did from there on, you can imagine, was biology of one kind or another. It was all about evolution from then on. But his training and his experience and his publications up until the publication of The Origin are befitting of any geologist. Now, here's what Darwin wrote. I do believe that natural selection will always act very slowly, often only at long intervals of time and generally on only a very few of the inhabitants of the same region at the same time. I further believe that this very slow intermittent action accords perfectly well with what geology tells us of the rate and manner at which the inhabitants of this world have changed. What did he know about how fast evolution was? Or was this just politics, this idea that it was very slow? Did that make it more palatable to be very slow? And after all, if you have as much time as you need, who could argue with you? Now, the outline for this talk is first, why study rates? Second, how to study rates. And finally, I want to come back to the question, how fast is evolution? Give me a number. And you'll leave today with a number. Now, to illustrate what Darwin knew, I want to start with The Origin of Species. This is really where he summarized 20, 25, 30 years of thinking about the question. He'd been around the world, he'd collected fossils and living animals all over the world. He'd had plenty of time to think about them, and this is the presentation of the thought. In those days, books weren't illustrated like they are today. How many illustrations did he publish in The Origin of Species? I like to tease biologists with this because, of course, they think of The Origin as a kind of Bible. And they don't always know how many illustrations were in it. And I wouldn't be worried about that except that there was only one. There's only one, and this is it. And it's an attempt by Darwin to give a picture of what was in his mind about how evolution took place through time. It's the only picture we have. The rest of it is all words. The bottom axis, I should say the vertical axis here, is time. And he specifies that this is in thousands of generations. So each of these units he imagined was about 1,000 generations up through time. Or he said maybe 10,000 generations. So we've got a pretty good idea what this axis means. Now, we're at a physics department here. Why would he put the independent variable on the y-axis? Physicists, mathematicians, the rest of us often put the independent variable on the x-axis. Why did Darwin stack t up through time? Because that's how stratigraphy works. He was a geologist. And my diagrams, too, will have time going up this vertical axis. But don't be fooled, it's still the independent variable. And we plot the dependent variable or variables on the other axes. But we stack this up through time because in our minds, that's how the layers go. Now, here's the problem with Darwin's diagram. This, he says, represents something about the form of animals. But it's not specified, and worse, it's not scaled. There's no scale. So this is some kind of difference. In my mind, I always imagine this is the small end of the scale, and these animals over here are larger. And each of these lines are diversifications, and then A few surviving and more diversification, a few surviving up through time. Some of the lineages change through time, some of them don't. So some persist all the way through, some become extinct without changing, some change and still become extinct. So this is the picture Darwin left us with. What did he know about how fast evolution is? Nothing, really, because this axis isn't scaled. He didn't try to measure anything. In fact, I think at that time he didn't have anything to measure. And so his idea that evolution was slow is really just something to make it more palatable. I think he believed it was slow, and of course evolution can be slow, but is that typical of it? Now I want to say a thing about rates, because I'm going to talk about rates here. And rates are just a little different if you've got time, the independent variable running up the y-axis here. Instead of thinking of a rate as the slope of the line, you have to think of a rate as the deviation from vertical. So here's a rate, a positive rate here deviating, another positive deviation. Here's a negative rate, negative deviation, positive, positive again. So we wind our way up charts like this. So it's really no more complicated than the slope, but it's just the slope as a deviation from vertical. Now, I got really interested in this when I was a student, because when I was a student, Steve Gould and Niles Eldredge published their theory of punctuated equilibria. And I expect most of you have heard of that. Their idea was that evolution isn't gradual step-by-step like Darwin diagrammed, or like my textbook when I was an undergraduate showed. Instead, they said, speciation takes place so fast we can't see it, and then animals are constrained not to change until, by some miracle, a change happens that we can't see, and then they don't change again. I once read him saying that human evolution is over. We aren't changing. Now, that was published in Cosmos magazine, which is a ladies' magazine, and I don't know how seriously he took the interview, but that's what he's quoted as having been told. I learned about this as a student, and I thought the idea was so nutty that I went back to the museum at Yale, went down in the basement, and spent a week measuring all of the Hyopsodus animals in the basement, for which we knew the stratigraphic position in Wyoming, and made this chart. Now again, time is going up the vertical axis. I don't know what time is. These are stratigraphic elevations above baseline here in feet. So I know we're getting younger as we go up through the stratigraphy, and I know that generally speaking, the time here is about 3 million years. So we can interpolate and get an estimate, if we want to, about what's going on from step to step in this sequence. On the horizontal axis, I plotted tooth size, measured as the length multiplied by the width, so the crown area of the first lower molar, and these are statistical samples, means and standard errors and standard errors and ranges up through time. And sent this to Nature, and they published it, and of course had a struggle with Steve Gould from then on because he didn't appreciate it. It didn't look like his punctuated pattern. To me, it looked much more like Darwin's branching pattern up through time. And Steve came back, Steve and Niles Eldredge came back and said, oh, you don't understand. We're not talking about the big picture here. We're talking about from this sample to this sample. That's a punctuation, then this is stasis, then that's a punctuation, then this is stasis, then that's a punctuation, then this is stasis. And I realized their theory wasn't scaled, and hence wasn't testable, and I dare say lost interest in it. But I realized that one way to tell the difference was to calculate rates of change. And the big mantra of Gould and Eldridge was, stasis is data. Remember all those vertical lines going up the page in their chart? Stasis is data. Well, here are the vertical lines. This is all what we call stasis, no change through time. You're going up vertical, no deviation, that's a zero rate, effectively. And so it looks like this on a chart. Here's 0. Now, I can't, I had to take an arbitrary angle here, an arbitrary measurement, and imagine what the positive deviations are out here in right field and the negative deviations over here in left field. And the key thing is that there's nothing in between. I've sketched here what you might expect, but the black lines show an actual numerical calibration of this, characterization of this diagram. And I did the same thing for Darwin's diagram. And sure enough, most of the lines going up the page are vertical. Stasis predominates. But about that are deviations, both positive and negative, and those pile up as a kind of normal bell-shaped curve here. And this led me to conclude that stasis doesn't distinguish the two models. You can argue stasis is data all you want, but that doesn't distinguish them. What would distinguish them would be an empirical distribution of rates. Will it look like this, or will it pile up like this? And in this way, the idea becomes testable. And so this changed my focus to evolutionary rates. Now, how do you study rates? I want to start by talking about a bell-shaped curve, and I always think this when teaching a big class, and I think it as I look at us here. I wish when we came in we could each register some number like our stature. And then I wish I had it all hooked up here so I could put up right now a histogram of our statures, or our tooth size, or the length of our femur bone, or something like that. Anything we could measure would be a bell-shaped curve. Now, where does a bell-shaped curve come from? How do you make a bell-shaped curve? If you want to make one at home, how do you do it? It's easy. You can do it flipping coins. You can do it easier, it turns out, rolling dice. If you roll one die, you've got an equal chance of a 1, 2, 3, 4, 5, or 6 coming up as the total count on the surface, on the top of the dice, on the top of the die. If you roll 2, that's what I've shown over here, this is the distribution. There are 2 ways you can get a 3, or there are 2 ways you can get a 2, and then there are more ways you can get a 3, and so forth. If you roll 3 dice, this is the frequency of the combinations. If you roll 4 dice, look at that. 4 dice. The frequency of the combinations from playing with 4 dice already gives you a pretty good bell-shaped curve. Now, these days we don't necessarily sit at our desk and roll dice. We're more likely to program a little simulation that will do it for us. And I've done that, and done it thousands and thousands of times. And of course, you get fascinated with it. It's fun to watch. And you can generate perfect, beautiful bell-shaped curves. Now, what's the message of that? If our variation is bell-shaped, what is that telling us? That's telling us that many small differences are adding up to make us what we are. It makes me crazy to read over and over again these days that somebody found a gene for this or a gene for that. Yes, if you knock out a gene, it will have a consequence, and it's usually pretty serious. Yes, you can identify a gene that is associated with a disease, but it's going to be a lot more complicated than that, because all this bell-shaped variation means there are many genes involved. So I'm going to talk about how you change a bell-shaped curve into another one, how you move it, in other words. Now, I like to tell my students something that I think is true. I've been teaching here 35 years. That's at least a generation. I think students today are taller than when I started. I walk across campus and I look at the boys and I look at the girls and I think they're taller. And so if you want to think about my generation to the next, here's my bell-shaped curve, and I've actually scaled this to be representative of my generation and scaled this to be representative of the current student generation. Remember, I was a student once too. And this is the difference. And I've modeled this so that I've actually built in something called heritability here, because to get from the mean here to the mean there, we're going to have to overshoot a little bit and then fall back, because all that we'll achieve by selecting to get here won't be heritable. Some of it will not be. I've assumed about 0.5 of it will be here, and then fall back to this. The whole trick is moving a bell-shaped curve. Now, I said from my generation to the next. That, I think, is the fundamental time scale we're interested in. Because evolution is never about me to my grandchildren. I can't get there except going through my children. It's always about one generation to the next. Going forward, going backward, however we think about it, that's the fundamental scale. Now, how do we talk about moving the mean from this curve to this one? What kind of units would be appropriate there? The early literature about evolutionary rates is in something called Darwin's, named by a biologist named Haldane, J.B.S. Haldane. And he defined these as change by a factor of E per million years. Oh boy. Does a factor of E speak to you? What's a million years? I mean, I know what a year is, and I can imagine what a million years is, but how's that related to getting from here to there? A million years is completely the wrong scale. What about factors of E? Well, we could do it that way, but it's not intuitive. These are logged, so yes, the units are factors of E somewhere deep down under there. What's the natural way to think about a normal curve? What are the landmarks of a normal curve? What are the parameters? Well, if we're talking about a standard normal curve, there are only two. There's the mean, where it's located, and there's the standard deviation, how dispersed it is, how broad it is. And the standard deviation turns out to be a very simple, easy landmark, because the standard deviation is just the inflection point. Notice when this curve comes down, it's concave downward until it gets here. That's the inflection point, and then it becomes concave upward. It's perfectly symmetrical. The same thing happens on the other side, and there's the standard deviation. So from the mean to that inflection point is a standard deviation unit. Same thing on the other side. And I've scaled off, the vertical lines here are standard deviation. So that's 1, that's 2, 3 would be right about where we go into the tail here. And we say we've got 95% of the variation under the curve here in this area, plus or minus 2 standard deviations. Or if you want 99% of it, you can go out plus or minus 3 standard deviations. You can eyeball this on any normal curve. You can eyeball it because it's just the inflection point. So that's a really simple, really natural way to talk about change. Now, when I modeled my generation, the stature of my generation, and the stature of the current student's generation, when I modeled that, I did it in standard deviation units. And it turns out the difference is about 1/10 of a standard deviation. Now, I didn't actually measure the students, and I didn't, of course, can't go back and measure my generation when we were students. But other people have. There is in anthropology a classical thing called the human secular trend. And that is over the last 3, 4, 5 generations, we have been documented to be getting taller through time. And the real study of it, the careful study of it, was by a man named Gordon Townsend Bowles. Got his PhD at Harvard and published this book from his thesis in 1932. New Types of Old Americans at Harvard and at Eastern Women's Colleges. And here's a graph from that chart, and it's the simplest one, and I'll just point out it has here time in decades on the abscissa where it should be, and stature on the ordinate. And so the slope of this line is the rate. And the increase is about 1 centimeter per decade, which turns out to be 1/10 of a standard deviation per generation. Again, per decade isn't going to help us in evolution. If we're talking about humans, that's about half or 1/3 of a generation. So translating this into the units we're interested in, that's about 1/10 of a standard deviation per generation. And that's where I got the number I just gave you, and that's how I made the model that I just showed you of the bell-shaped curves moving. So keep that number in mind, 1/10 of a standard deviation per generation. Some of you may know Stanley Garn, who was a professor here for many years, an expert in this kind of thing. I used to ask him, can that be evolution? Oh, no, it's too fast. Oh, how fast is evolution? Now, what happens when we do experiments? Because, of course, we can go in the genetics lab, we can go and we can breed fruit flies the rest of our days, and we can do lots of experiments, and we can select them to become larger, we can select them to become smaller, we can change the venation pattern, we can do all kinds of things by artificial selection ourselves. My favorite is a mouse experiment that proceeded for 23 generations, raising mice, selecting them to be larger, selecting them to be smaller, and the geneticist who did it, a man named Falconer in Scotland, succeeded in making mice twice the size they were when he started, and succeeded in making lines of mice half the size they were when he started. So we can do it. And when we do it, we get rates of change over here. Now, these are in standard deviations per generation, and the axis is a logarithmic axis base 10, so 10 to the 0 is 1. So some of these changed. A few of the steps between generations were as much as a whole standard deviation. Mostly, though, they cluster around about 1/10 of a standard deviation per generation. You heard that number before? Yeah, that's what I was talking about with human stature. Now, geneticists are, or at least used to be, a modest crowd, and they would be the first to tell you, oh, this can't be evolution, it's too fast, it's something I did in the lab. It's something I did. It can't be representative of nature. It's something I did. Okay. Now, Darwin's experience was with fossils changing through time, even if he didn't actually quantify this. And certainly my experience is with fossils changing through time. And I've compiled a big histogram here of rates of change, again in standard deviations per generation from the fossil record. And these are all down at about, what's that, a 10,000th or a 100,000th of a standard deviation per generation. And this kind of rate, based on fossil horses, was first done by Haldane in 1949. And even he said at the end, this is so slow, probably isn't important, it can't be important. And Russell Landy, a famous geneticist, and Michael Lynch, another famous geneticist, have done similar treatments of this kind of rates from the fossil record and concluded these are so slow they can just be random genetic drift. There's nothing about this that would ever get you anywhere. So that's too slow. Well, the rates we saw were too fast. These are too slow. Must be somewhere in between. Now, I want to digress just for a minute to tell you something about how a paleontologist calculates rates. Here's a sequence of fossil horses up through time. This is just like the Hyopsidas, except now I have an age model that's calibrated based on the magnetochrons out of the section. This is 55 million years ago. This is 55. This is 50. Where are we here? 55, 54 is here, and the top is 53.4. So this is about 1.6 million years, something like that. And we can interpolate, and we can estimate from the size of the animals the generation time was a year or two. And so we can figure out how many generations are in this. And we have good samples. Each dot here is an individual specimen. You can see they zig and zag up through time and shift and come back. We can calculate the rate from this sample to the next, to the next, to the next, to the next, to the next. And when we do that, we get a distribution of rates. These fossils here, 469 fossil horses, represent a lot of work. I started, when I came to Michigan, taking teams of 8, 10, 12 students to Wyoming for the whole summer. Summer after summer after summer. Now, in the process of that, we collected these fossil horses. We, of course, also collected lots of other things we're interested in. I could never manage that on my own. And so I want to come back to the museum here in the background as a very important institution that makes research like this possible. It provides the launching pad for the expeditions. It provides some staff for managing the collections when they come back. If you go from here to the museums today, you can have a behind-the-scenes tour. Down in the basement, you'll see rows and rows of cabinets where these specimens are conserved, so that if somebody else wants to measure them or measure something different, they can. If they doubt what I did, they can come redo it. It's archived there. And that's thanks to the museum as an institution to make this kind of research possible. So the compilation of fossil rates that I showed you are clusters like this plotted on as a histogram. Now let's come back to the main story here. The experimental rates are too fast. The fossil rates are too slow. Well, what do we have that's in between? Well, we can try something that is on a natural time scale, but a fairly short one. So we can look at what I call historical rates. A nice study was done by Philip Myers, a professor of biology here, again through the museum, where he looked at rates of change, or differences, I should say, between rats on the Galapagos Islands. Now, most people don't go to the Galapagos Islands to see the rats. They've been there 300, 400 years, ever since Portuguese sailors first discovered the Galapagos, and the rats got off the ships and stayed. And each island has its own rat fauna. And we know they diverged now for 300, 400 years. That's a lot of rat generations. And you can sample each island and measure the differences between the islands. And then knowing that those accumulated over 300, 400 years, you can figure out rates. Or you might study sparrows in Ann Arbor today and compare them with sparrows from Ann Arbor at the turn of the last century, 100 years ago, and calculate rates of change from that. And that, again, depends on the archiving in the museum. And if you do that, you get a distribution of rates like this. Well, these are in between what we had before. That's good. They overlap on both ends. Is this more reasonable? Is this what we should assume? All these rates. How fast is evolution? Give me a number. Now, sometimes we do things that surprise us, and what I'm going to tell you from here on is based on one of these surprises. Some of you are alumni and will remember something called the Michigan Terminal System. We used to have deck riders scattered all over, and we had one in the museum. We could hook up to the central computer, and we could put our data in on punch cards, and then we could sit in the office or in a computer room back in our own building and work on the data. And I was doing this with evolutionary rates, a smaller data set, but same idea. And just by chance, I made a plot of the log of the rate versus the log of its denominator, the interval of time over which that rate had been measured or calculated. And I got a big surprise because it was a perfect inverse relationship with a very high coefficient of determination here. This number means that 92% of the rate we observe is due to the denominator. Oh, for goodness sake, I did all that work to divide all these numbers by the time over which they were measured to make a rate, and they still depend on the time. What's going on here? Now, have any of you ever seen this kind of inverse relationship on a log-log plot before? There's a really interesting study in Science in the 60s by a man named Mandelbrot, 1967, where he uses the coastline of Britain as an example of a new word that he introduced, fractal. And we usually think of fractals as some kind of wild, pretty diagrams or something. A fractal is actually something much simpler than that. Fractal is just a contraction of fractal dimension. It's about dimension. It's about points and lines and planes being 1, 2, 0, 1, and 2 dimensions in a normal Cartesian space. But it's about there being dimensions in between. There's something in between being a line and being a plane. And the coastline of Britain here has a different length depending on how you measure it. It's easy to imagine. If we come up to the screen here and put a meter stick on it, it's going to be about a meter long. If we use a shorter ruler, maybe a centimeter ruler, it's going to be longer. I think you can see that. You're going to get more of the detail. And if you finally go to the absurd extreme of going out on that coastline yourself, you're just going to be confused because you're not going to know which rocks to go around, and it's going to get so complicated that effectively the length will become infinite. The length depends on how you measure it. Now, that doesn't help unless you figure out a way to manage this, and Mandelbrot did. Here he's plotted the length of the west coast of Britain on a scale of measurement and shown that the length decreases systematically as you measure it with a longer and longer ruler. And this he calls the fractal dimension of the coastline. He says it's not a line, it's not a plane either, it's something in between. And he quantifies it by noting that the slope here is negative 0.25. He introduced the convention of taking the absolute value of that, so forget the minus sign, 0.25, and add 1 to it, and that will give you a fractal dimension d of 1.25. 1 would be a line, 2 would be a plane. So this is something in between, and that's where it is on that scale, 1.25. It's a quarter of the way to being a plane. Now, I bet every one of us can remember being in primary school and coloring a diagram or a flag or something. And we had crayons, with which you can make lines, and we were expected to color an area. Remember? And we did it. We made lines that were so complicated, so rough, back and forth so much that they filled the area. That's a line with a fractal dimension approaching 2. We made a line so complicated, we filled a space with something, with a line so complex, it was virtually a plane when we got done. And that's exactly what the teacher wanted. Now let's come back to this. The problem is we have all these rates, and we don't know what we're interested in yet. A colleague of mine, Fred Bookstein, some of you know, 20 years ago, when he saw the first plot like this, jumped to the conclusion that this means evolution has no rate. And I thought about that, and I thought, no, it means evolution has many rates. They just depend on the scale. What Fred meant is that there's no singularity, there's no rate to choose of all the rates that are there. And I thought about that, and I thought, no, there is a rate to choose. And that's the generation to generation fundamental scale of the process of natural selection and evolution. Generation to generation. Now, where is that on this diagram? This is a log-log scale, remember. This is like Mandelbrot's plot of coastlines. Well, 10 to the 0 is 1. 1 generation is the intercept of this plot. That's handy. It turned out nice. What we're really interested in is what's happening on a 1 generation time scale. Now here are all these mouse selection experiments that Falconer and others thought were too fast to be evolution. Everything about this distribution points to that intercept. That stuff's not too fast. Even my fossil horses down here point to evolution on a generation-to-generation time scale being on the order of 1/10 of a standard deviation. Now we're started, finally. So this isn't unreasonable, what we saw in the secular trend of our stature increasing. It's pretty shocking to think that the difference between my generation and my students' generation could be evolution. But the more you think about it, you think, why not? So the number I would point to is about 1/10 of a standard deviation per generation. The important thing is that's on a time scale of 1 generation. That's a rate measured here. All of these other rates we can explain as the same thing averaged as they zigzag back and forth over time. And you saw in my fossil horses, they zigzag back and forth over time. Now, what does a slope, here we've got the equation of this line, what does a slope of negative 0.92 mean? That's the slope down through here. That's like the regression of the coastline length on its denominator on the scale. 0.92, take the absolute value of that, add 1, 1.92. That's a fractal dimension of about 2. That's like using a crayon to fill an area. That's zigzag back and forth. Can it be that evolution mostly goes back and forth? Can go somewhere fast for a short time, but can't go very far? Does that make sense? Well, we can do modeling to model this. Here's a random walk, just to give you a baseline, a random walk. And here I've calculated every single rate between every pair of points for 200 steps of this random walk. I don't even remember how many that is. It probably says somewhere. It's about 20,000 or something like that, or maybe more. And they're all shown here. Some are positive, some are negative. Here I've treated those the same by thinking, well, the positives and the negatives are two experiments. They don't, the sign doesn't matter, in other words. So here I've piled them all up together. I've effectively folded the negative ones up on the positive ones. I've taken the absolute value. And here I've done a log-log plot of those. And here's a plot with a slope of negative 0.5 approximately. It's actually negative 0.6 here, but it's close to negative 0.5. And that turns out to be the expectation for our random walk, a fractal dimension of 1.5. So that's really easy. Now, we can constrain the random walk in a different way and make it so that every step is positive. Yes, we sample from a distribution of rates, but we make the sign positive every step. And so, of course, it will always go in one direction. It may wiggle, but it'll always go in one direction. And if we do that and look at the rates, it'll have a fractal dimension of a slope of 0, and add 1 to that, a fractal dimension of about 1. And that just tells you this is pretty much a straight line. It's pretty smooth line, I should say. It's not quite straight, but it's smooth. And we can do the opposite and constrain every step to change sign. And that's like our coloring experiment again. Back and forth, back and forth, back and forth, back and forth. And where's that going to go? That's going to march down the page with a slope of negative 1. Absolute value, add 1, fractal dimension of 2. So this kind of framework gives us an easy way to interpret these rates. So I mentioned the fractal dimension 1.92 is about 2. So what that tells us is this time series is filling an area. It's what we call stasis. Now, to understand how evolution can be so dynamic and so active and not go anywhere, I want you to follow through with me on this thought experiment. We're mammals. The smallest mammal is a least shrew, and they crawl in burrows that are about the diameter of a pencil, just to give you a sense of the scale. The largest mammals are great blue whales, and they weigh something like 100 tons. They're enormous. But there's never been anything bigger, and there's never been a mammal much smaller than at least Shrew. Why not? What happens if you get smaller than at least Shrew? Has anybody ever tried trapping shrews? They're in your garden. If you do, you better pay attention, because those poor guys will die in half an hour or an hour if they don't eat. If they're exposed to the atmosphere, not in their burrows where they can keep warm, if they're exposed in a trap and aren't fed, they'll die. The physics of their heat loss, surface to volume constraints, means they die. You can't be smaller. Same thing with the great blue whale. You know, it doesn't live on land. There's a reason for that. It's too big, couldn't support itself. It's as big as it is, and there aren't any mammals bigger because they can't find enough food. So there are constraints. Now, I'm going to put this into a context of tenths of a standard deviation equivalent to a generation. That's our rate here. That's the number we've learned. Tenth of a standard deviation equivalent to a generation. So I want the difference between a shrew and a great blue whale to be expressed in tenths of a standard deviation. Now I'll just say, if you measure blue whales and measure shrews and measure other mammals in between, put them on a logarithmic scale so the standard deviations are comparable across the whole scale, and ask how far apart are they in standard deviation units, they're about 100 standard deviations apart. That's 1,000 of my tenth of a standard deviation units. So that's 10 to the 3 units. Now, they diverged in evolution about 55 to 60 million years ago. Oh, that's a long time. How many generations is that? Well, for shrews, they probably have several generations. They do have several generations a year. Great blue whales are like us. They have 20 to 30-year generations, maybe longer. So if we again take something in the middle and ask how far have we come in time since whales and least shrews diverged, we get an answer on the order of 10 to the 7th generations. Now we're all used to playing board games and games like chess, and we're used to thinking in terms of square boards here. What's the board on which, if you think of evolution as a game, on which this was played for the shrews and the whales? Well, this side is going to be 10 to the 3rd. This is the difference between shrews and whales in standard deviation, tenth of a standard deviation units, 10 to the 3rd. And this side of the board is going to be time. Since they diverged, that's going to be 10 to the 7th generations. Oh boy, what kind of board is that? That's not square at all. That's 4 orders of magnitude longer than it is wide. So we can change at a tenth of a standard deviation per generation. We can change a shrew into a whale. We can imagine changing a shrew into a whale, at least a shrew-sized mammal into a whale-sized mammal in a few thousand generations, maybe not that. Cross the whole space and back again. And here I've tried to diagram this thing, and even here I can't get it on the paper. Think of a tube that's 10 centimeters at the top and a kilometer long, like an oil well or something. That's the space in which evolution has taken place, and that's just the evolution of mammals like us, has taken place on land since the age of dinosaurs. So we're very constrained morphologically, and geological time is very long. Now, that helps explain why adaptation is so rapid when selection is powerful, and of course it is if it can move things at rates like a tenth of a standard deviation per generation. But geological time is very long. And environmental perturbations, while they're happening more or less all the time, are still relatively rare. So I think that's why the fossil record looks punctuated. It's rapid response to punctuated environmental change. It's not that the process of evolution itself is punctuated. So finally, if we come back to Darwin and his idea that evolution acts very slowly, that was very palatable, but I think that's one thing he got wrong. Now, the interesting thing about it is, what does this say about natural selection? I've already alluded. If evolution is this fast, natural selection is very powerful, much more powerful than he represents in this quote. So I think he got the important thing right. Now, how fast is evolution? Just to conclude, I would say all evidence points to an average of about 1/10 of a standard deviation per generation on a time scale of 1 generation as being representative. I think that's the rate we want to stick in our mind, and that's very, very fast. That means that closely related species, and these often differ by only 3 or 4 standard deviations, may take only 30 to 40 generations to separate, to speciate. That's fast. And for small mammals, that may take only a few years. No wonder evolution is ubiquitous, inescapable, and fundamental for understanding us as humans, the diseases that plague us, and our integration and dependence on the biosphere we live in. Thanks for your attention.
Saturday Morning Physics is sponsored by the M. Lois Tiffany Endowment, the Hideko Tomazawa Endowment, the Physics Department, and by gifts from friends of the program.